ExiPrep 16 Plus is an automated nucleic acid extraction system capable of processing up to 16 samples simultaneously. Based on magnetic bead technology, it enables nucleic acid extraction from a wide range of sample types, including blood, bacteria, and plant tissues. This is the exterior and internal layout of the ExiPrep 16 Plus. On the front of the instrument, you can find the power button, touchscreen, and door. When the door is opened, the base plate is visible. The base plate can be moved forward and backward by holding the handle. When the base plate is pulled out, the internal structure can be seen. From the front to the back, the base plate is designed to accommodate the disposable tip rack, Elution tube rack, buffer cartridge 1, waste tray, and buffer cartridge 2. Before using the instrument and kit, please check that all kit components are properly prepared. Confirm that all required components are included and that the quantities are correct. Once the components have been verified, we will take a closer look at the buffer cartridges. The first two rows at the front of buffer cartridge 1 contain proteinase K. This is where the sample is loaded. The sample is then transferred sequentially through the binding buffer, magnetic bead solution, and binding buffer again for nucleic acid extraction. Buffer cartridge 2 is used for the washing and elution steps required for DNA extraction. Once elution is complete, the extracted nucleic acid is transferred to the elution tube located at the front of the base plate. Before use, check that the beads and reagents inside the cartridges have settled properly. If necessary, gently tilt the cartridge from side to side to allow the beads and reagents to settle. After confirming that the reagents have settled, open the cartridge lid, perform punching, and proceed with sample loading. The punching positions and number of holes should be adjusted according to the number of samples. If there is 1 sample, punch the cartridge as shown. If there are 8 samples, punch the cartridge as shown. If there are 16 samples, punch the cartridges as shown. After punching, use a pipette to load approximately 200 to 300 microliters of sample in a buffer cartridge 1. Close the lid to complete cartridge preparation. Turn on the instrument by pressing the power button on the front panel. Touch the Press to Start screen on the LCD to begin booting. Once the instrument has finished booting, the cartridges can be loaded. Pull the base plate forward using the handle, confirm the numbering, and load each component into the correct position. Insert the waste tray into the space between buffer cartridge 1 and buffer cartridge 2. Next, load the elution tubes into the elution tube rack. The elution tubes should be placed according to the number of samples and the punching positions. When using multiple samples, ensure that the tube caps of the elution tubes do not overlap. As shown, the tube caps should face outward. After placing the elution tubes, insert the disposable filter tips into the disposable filter tip rack. The disposable filter tips should also be placed according to the number of samples and punching positions. Hold the base plate handle and push it into the instrument until a clicking sound is heard. Close the instrument door. On the menu screen, touch the Start button. On the Prep Setup screen, enter the appropriate protocol number. For the K4211 ExiPrep Plus Blood Genomic Kit, enter protocol number 101. Once the protocol number is entered, the prep type and sample source will be set automatically. Confirm that the prep type is set to genomic DNA and the sample source is set to whole blood. If the settings are incorrect, press Clear. If the settings are correct, press Enter. On the Prep Setup, select Elution Volume screen, confirm that 50 microliters is selected, then press OK. Once all settings are complete, review the final checklist. After confirming all items, press OK. On the Running Mode screen, press the Run button to start the automated nucleic acid extraction process. When the extraction process is complete, the work completion screen will appear. Open the instrument door and pull the base plate forward using the handle. Close the caps of the elution tubes and collect the extracted DNA. If the extracted DNA will be used in the short term, store it at 4 degrees Celsius. For storage longer than 1 week, storage at -80 degrees Celsius is recommended. After sample recovery, collect all used components, including the cartridges. Dispose of the waste liquid remaining in the waste tray, use cartridges and disposable filter tips according to appropriate disposal procedures. Clean the collected waste tray and base plate using paper towels and 70% ethanol. After cleaning, push the base plate back into the instrument and close the door. On the work completion screen, press OK to initiate UV sterilization. UV sterilization will run for 15 minutes. Once UV sterilization is complete, turn off the instrument using the front power button. This video is intended for SOP training purposes for the ExiPrep 16+.